Okay, so let's talk about how viruses work and their life cycles. So the first thing that happens in a DNA, so this is very specific to DNA. We'll talk about the RNA one on the next slide. But a DNA type of virus, first thing that happens is that virus, so you'll see this picture, it has a protein holding around the outside and then DNA strands in the middle, and that's pretty much it. So the virus attaches to the host cell and injects viral DNA into the host. So this virus cell, when it hits the cell membrane of a cell in whatever kind of human or whatever kind of organism that it can kind of unlock, you'll see that one of these little spikes on the side. So you'll hear with coronavirus a lot of spike proteins. That little spike acts as like a key, and it unlocks the outside and inserts itself into the cell or the host cell. Then the virus uses, so it unlocks itself, the protein becomes part of the protein on the outside of the cell, that membrane, and then the DNA on the inside travels into the nucleus, and the virus uses the host cell's machinery to replicate its DNA. So that DNA gets into the DNA of that cell and starts replicating, just because those enzymes in that host cell don't necessarily recognize that that is foreign DNA. They just see the DNA and start replicating it. Then genes on viral DNA are transcribed and translated by host cells machinery to form viral proteins and mRNA. The large viral protein is chopped up into smaller active proteins by a specific viral protease. So the specific viral protease is an enzyme that the DNA codes for. It's made through all of our same protein synthesis steps that we've seen before. And that viral protein is chopped up into smaller active proteins. So the protease, it takes the protein that's formed and it's inactive. And if you have that viral protease that's around, so that enzyme that the cell or the DNA from the virus makes, it will chop it up into proteins that can be used by the virus to make more virus cells. So you'll also replicate the DNA. So that DNA that gets replicated and all of the new proteins and everything, those go towards the cell wall. Once there's enough of those kinds of proteins together, it initiates sending out the new genes. So viral proteins are assembled into new virus particles. So inside the cell, those proteins and those new replicated DNA strands will all form into a little ball. And the cell synthesizes so many virus particles that it eventually bursts and viruses are released. So you get a lot of virus particles within that cell. You're going to burst open that virus cell and release a lot more of those viruses. So it will exponentially, this process will exponentially occur. So once you get enough, you burst open, it attaches to, let's say, 50 more cells, and then 50 cells within a couple hours will do that again as well, over and over and over. So RNA type viruses are called retroviruses. So retroviruses, so HIV is an example of this, contain RNA instead of DNA, and they require a process called reverse transcription in order to for them to replicate. So let's look at this life cycle. We have a retrovirus, which contains viral RNA, but no viral DNA enters the cell. Same thing, virus comes into the cell. Now you have your viral RNA. This is also inside the RNA virus that goes into the host cell. There also has something called reverse transcriptase. So what this will do is it will take viral RNA, that enzyme will then go backwards and create a strand of DNA from the RNA. Then you have so it's kind of like a backwards process. That's why it's reverse transcriptase. You take the RNA, make a strand of DNA, then that DNA can be replicated. So now you have a full strand of DNA. So then once you're inside that cell and have all of the materials to make, so you need all of those nucleic acids. The virus doesn't have the nucleic acids. You can make viral DNA. The viral RNA, okay, I already said this part. Viral RNA uses reverse transcriptase, a polymerase enzyme included in the virus, to produce viral DNA strands. The viral DNA strands uses the host machinery to form complementary DNA strands. The double-stranded viral DNA is called a provirus. So after it is replicated inside a cell, then it is called a provirus. The viral DNA, we're up at number 3 right now. The viral DNA, or the provirus, inserts itself into the host chromosomes. This is catalyzed by the enzyme called integrase. So this DNA, kind of like a plasmid, then goes into the nucleus of the cell where all of the chromosomes, all of the DNA is for that cell, and it inserts itself into the DNA for that particular cell. That integrated viral DNA is transcribed and translocated into a large viral protein by the host machinery. So then as the host cell's machinery replicates the DNA, it will make more and more of the viral RNA, the mRNA, and that will again make packets of the new virus that can then be sent out into to infect more things. So overall, these are general schemes for how these work. There are differences that can occur, and you'll see that there's slight differences with like coronavirus, COVID-19, and how they found it works. But these are the general life cycles, but overall they act very similarly in that the virus comes in, uses the machinery of the host cell to make more virus, and then that virus will go out and infect more things.